The topic is pH meter. So pH is defined as concentration of hydrogen ions in the solution. The pH expressed as minus logarithm of hydrogen ion. So pure water has pH nearly equivalent to 7, which is considered normal. Solutions having pH greater than 7 are considered basic, whereas those lower than 7 are called acidic. So pH is defined as concentration of, and the formula of pH is minus logarithm of hydrogen ion. Neutral pH is 7. And acidic and basic pH? Less than 7 is acidic, more than 7 is basic. So method of pH measurement. Conventionally, pH was measured with the help of litmus paper, which determined whether the solution was acidic or basic. Later on, pH papers were used, which gave subjective range and were not precise. Nowadays, pH is measured with the help of pH meter, which gives precise pH measurement. So by litmus paper we can detect pH, na? Like if it is acidic or basic. In litmus paper, if it is acidic, then
blue turns red.
This is red. Blue turns red. So pH meter. The pH scale is a series of numbers that express the acidity as the concentration of hydrogen ion in the solution. The normal hydrogen ion concentration in terms of gram per liter is in the range of 10 to the power minus 1 to the 10 to the power minus 14. So this is the pH meter. This pH meter, the same pH meter we have in our lab. So principle, what is the principle of pH meter? pH meter is based on the principle of potentiometry. What is potentiometry? Potentiometry is measurement of electric potential generated between two electrodes of the electrochemical cell due to difference in hydrogen ion concentration. So the principle of pH meter is potentiometry. So if a metal plate is placed in a solution of its own solution, it loses ions into the solution and itself becomes negatively charged as compared to the solution. This generates an electrical potential on the metal plate or the electrode. So if two different metal electrodes are connected in this way, the difference in their electrode potential can be measured as electric potential. Hence, if one of the electrodes is a standard electrode, the electrode potential of the other can be measured by comparison. Electrochemical cell consists of two metal electrodes, each of which is dipped into suitable but different solution connected by wire. The circuit is completed by potassium chloride or agar bridge between two liquids. Each metal and the electrode and the solution in which it is immersed constitute a half cell. What is half cell?
Half cell, one with positive polarity.
One in a solution where composed of an electrode and with an electrolyte.
Okay.
This constitute half cell. So if one of the half cells is arbitrarily assigned a fixed potential, then the potential of other half cell may be determined relative to it. If one of the half cell is assigned as fixed potential, then other half cell may be determined relative to the first half cell. The electrode, the potential of which is fixed, is called standard electrode and the other electrode is called measuring electrode. The electromotive force EMF is defined as maximum difference in potential between two electrodes obtained when the cell current is 0. So what is electromotive force? Electromotive force is defined as maximum difference in potential between two electrodes obtained when the cell current is 0. So components of pH meter. One is glass electrode. It consists of very thin bulb about 0.1 millimeter thick blown onto a hard glass tube of high resistance. The bulb contains 0.1 mole per liter hydrochloric acid connected to a platinum wire via silver-silver chloride combination. So this is glass electrode. The another one is calomel electrode. It consists of a glass tube containing saturated potassium chloride connected to platinum wires through mercury-mercurous chloride paste. When the electrode is dipped in a solution, the thin glass membrane separates two solutions of two different pH values. So, this generates potential difference across the membrane due to transfer of hydrogen ion through glass. The electromotive force of electrochemical cell gives measurement of hydrogen ion concentration. Recent instrument gives measurement by digital display of pH reading. The next one is combined electrode, which is actually seen in our pH meter instrument. In actual instrument, in pH meter, combined electrode is seen. A combined electrode having both reference electrode and measuring electrode is used. So the reference electrode is calomel electrode and the measuring electrode is glass electrode. The pH of unknown solution at 25 degrees Celsius is given by pH equal to E minus K by 0.0591, where the K is constant and E is the measured potential. So the standard temperature for pH is 25 degrees Celsius. If the temperature rises, then what will happen? In increased temperature, what will be the changes in pH? If in solution there is more number of hydrogen plus ion, so this solution will be acidic. So when temperature increased, there will be more dissociation of the solution. Yes, so there will be more H plus ion. So when the temperature increased, pH decrease. Same, when the temperature decrease, pH increase. Like at 25 degrees Celsius, the water pH of water is 7. But at 30 degrees Celsius temperature, the pH of water is 6.92. So when the temperature is raised, then the pH will decrease. pH will be more acidic. So the instrument is turned on and allowed to stabilize for 10 to 15 minutes. During this period, the electrode bulb should be kept in distilled water. After the instrument is stabilized, wash the electrode bulb with distilled water, wipe dry with filter paper and place it in a standard buffer solution. The choice of buffer is dependent on the pH of unknown solution. After calibrating the pH meter, wash the electrode with distilled water, wipe with filter paper and place it in an unknown solution. After the pH measurement, knob should be maintained in standby position. Wash the electrode with distilled water and place it in a beaker containing distilled water before turning off the instrument. The advantage of the glass electrode is that it can be used in any solution not being affected by organic compounds or oxidizing or reducing agents. A small quantity of the solution is sufficient for the determination of pH. Special glass membrane is required when the pH of the solution is very high, which is more than 10. Anything you have understood? Anything? Nothing. Okay. So, the components of pH meter are, previous slide, components of pH meter are calomel electrode and combined electrode. So this is the principle of pH meter. Okay. So this is the glass electrode. Glass electrode. This reference electrode is calomel electrode. Measure this glass electrode. This is measuring electrode, and this is reference electrode, calomel electrode. So in glass electrode, the tip contain silver chloride. So the glass electrode tip contain silver chloride, and the calomel electrode tip contain mercuric. We have written na mercurous chloride paste. And the glass electrode, the solution is filled with KCl solution, 7 pH. The pH of KCl here is 7. So why the pH is 7? This is neutral, na? Neutral pH. How it is 7 pH? What it means? The pH 7, neutral pH means what?
It's a salt.
So 7 pH neutral means they have same amount of hydrogen ion and same amount of hydroxyl ion. So it neutralizes itself. Okay. Next is applications. The use of pH meter. This is the last slide. So what is the use of pH meter? To measure pH of biological fluids. There is one table, pH of various biological fluids are cerebrospinal fluid 7.3, blood pH is 7.38 to 7.42, gastric juice pH is 1.5 to 3.5, lymph same as plasma, and seminal fluid 7.1 to 8.0, and bile pH is 7.5 to 8.8. So to measure the pH of biological fluid, the first point. Second point is to adjust the pH of buffer solution, which are used in enzyme assays. Next is to adjust the pH of various reagents used in biochemical assays. And fourth is clinical application includes measurement of pH of blood gases, carbon dioxide, oxygen and bicarbonate using blood gas analyzer. So there are few questions. Define pH. What is pH? So pH is defined as concentration of hydrogen ions. So is pH of pure water changes with increase in temperature? If there is temperature change, there will be change in pH. Yes. In more temperature, if the temperature is more, then there will be more dissociation of ions, na? So there will be more H plus ion if the temperature rises. So pH change with temperature. If temperature increase, pH decrease. If temperature decrease, pH increase. Why? There will be more number of hydrogen ion. More number of hydrogen ion. कैसे वो कैसे how? हाँ तो if one solution there is more number of hydrogen ion ना, तो this will be more acidic. If there is more number of hydroxyl ion, so this is more basic, na? So one number is more, na? If hydroxyl ion or hydrogen ion, hydrogen number ज़्यादा होने से acidic होगा, कम होने से hydroxyl ज़्यादा होने से और hydrogen कम होने से वो basic होगा.
Change color.
Yes. So in universal pH indicator, what is universal pH indicator? So this is a combination of various indicator in one solution or in a strip, like methyl red, thymol blue, bromothione blue, bromocresol green. So these are the various indicators. So these all are available in one strip or one solution. So this shows like changes in color, like red to green, red to orange, orange to green, green to blue. So in one strip everything is available. For every pH there will be one indicator, one color changes. So this is universal pH indicator. So what is the principle of pH indicator? Potentiometry. And use of pH meter?
Enzyme, biological acid.
To measure pH of biological fluids. Then to measure pH of reagents using biochemical assays to measure the pH of buffer solution. So what is buffer solution? Yes. pH is constant. For if we add minimal acid or alkaline, the solution pH will be constant. So this is the buffer solution. Then what is ABG analysis? So what is ABG analysis? Arterial blood gas analysis. The last point we have written now in application of pH meter. What is the last point of application of pH meter?
pH of blood gas.
Yes, pH of blood gases. So what is ABG? Arterial blood gas analysis. Arterial blood gas analysis. This is the test. This is the blood test to calculate the partial pressure of carbon dioxide, partial pressure of oxygen, and the arterial pH of blood and the arterial oxyhemoglobin saturation. These four we can measure by ABG analysis. Arterial oxyhemoglobin saturation. How we do ABG analysis? By blood test, na? Blood test. So what is the preferable artery for ABG analysis? Idea? Any idea? Radial artery. Radial artery is the preferable. The next is brachial artery. First one preferable artery is radial artery. Second is brachial artery. Then third is femoral artery. Why radial artery? For this instrument na, to test. So like in angiography, there are same radial artery is preferable. Why? So for some procedure, some artery is more comfortable to do the test. So more, that's why more preferable, na? So femoral artery, it's tough to do in femoral artery. So this is the less preferable. Understood? Okay. So we'll perform this test, the pH measurement in pH meter in our lab. Then we will see how to measure the pH of any solution. Okay? This is pH meter. So first we have to start the button here, na, on and off. There is one switch, on and off. Then there is 3 mode. pH. This is now on standby mode and millivolt. pH standby millivolt. So first we have to set in pH mode because we are measuring pH. pH mode. Now there is 2 buffer. One is pH buffer 7. This is neutral pH buffer. Another is pH buffer solution 4. This is acidic pH buffer. So there is 2 set button, set 7 pH, set 4 pH. So first we have to wash it in distilled water. Then dip it in
neutral pH.
Dip it in neutral pH 7 pH. Why? This is the first button is 7 pH, na? So first we have to set in 7 pH. This is 7. Now press this switch for little longer and leave it. Done? Now, so it's set. Now wash it, again wash it. Wipe it. Setting pH 4. 4. Now set. Done. Again wash it. So this is the electrode bulb. Again wash it in, dip it. This is our unknown solution. So this solution we have taken sodium hydroxide. Sodium hydroxide. So what is the formula of sodium hydroxide? NaOH. So the molecular weight is
40.
how?
17.
23 plus 16 plus 1. Total 40. Okay. So come here. Concentration equal to
Number of mole by volume.
number of mole by volume. So number of mole is, so this sodium hydroxide pellet, how did we prepare? With 500 ml of distilled water, we have taken 1 gram of sodium hydroxide pellet. Like available as this one tablet size na pellet. So 1 gram of pellet, sodium hydroxide pellet. So one is the weight is 1 gram divided by the molecular weight 40. 1 by 40 equal to 0.025. Divided by volume is 500 ml na. So in liter this is half. 0.025, then 2 comes in and 0.05. Concentration equal to 0.05. We can write this same as 5 into 10 to the power minus 2. And we take hydroxyl ion. Na? This is strong base. So we are taking hydroxyl ion OH minus equal to 5 into 10 to the power minus 2. So the formula of pH is minus logarithm of the hydrogen ion. Same for hydroxyl ion. POH equal to minus logarithm of the value of OH 10 to the power minus 2. So 2 comes in first, 2 minus log 5. So this is approx 1.69. So this is the value of POH. What? So value of POH is here what?
0.69.
So total value is POH plus pH equal to 14. So the value of pH is 1.69. So minus, what is the value of pH? 14 minus 12.3. Yes, 12.3. So the estimated pH will be 12.3 for our unknown solution, the solution for sodium hydroxide. Okay. One instrument, there are always some error. Na? For any instrument, there is 5 to 10% error. So, but the solution, this solution is prepared for very long, na? Before lunch. In morning, this solution prepared. So, when one solution is exposed in air, so it comes in contact with carbon dioxide, so it becomes little bit acidic, na? Because of the environment. So, it's coming 11.37, near about 12. But because of its exposure to air for the longer period, it's become little acidic. Okay. So why we have set this in 7 pH, 4 pH? Why? What is the use of 7 pH, 4 pH?
7 to neutral. 4 to acid.
Why we have to set with buffer? This is not standard. See, actually, in these bigger labs, na, they have pH meter of containing all pH, 14 pH. But this is a smaller instrument, na. So they have provided 7 pH and 4 pH. The purpose of setting pH is to, what is the function of buffer is?
Set the pH.
To maintain the pH, na. So that's why first we have to set this buffer with these 2 buffer, na, to avoid the error in the pH of this solution. So this is the purpose of setting pH. There is like no significant of this 7 pH or 4 pH. Okay. The purpose is to avoid the error of this pH. So we are calibrating with the pH meter.
So that the solution don't change the pH.
Yes. So that the unknown solution don't change the pH. So we are calibrating with this solution. Okay, with this solution we are calibrating with pH meter. Why? Like, what is calibration? Calibration means we know the pH of this solution, but the pH not set in instrument. So we are checking the preciseness of the result. So this is the calibrating with the instrument, calibrating with the pH meter. Calibration of the pH meter is for any solution like this, we can calculate the pH and we can calibrate with the pH meter if the result is precise or not. Okay, for any solution, if you dip this electric rod, electric bulb, this will give the pH of that solution. So what is this? This electric bulb. In principle of pH meter, we have shown there is two electrode. What are those?
Standard electrode, measuring electrode. Standard.
One measuring electrode, one is reference electrode.
Standard.
So one is measuring electrode is glass electrode. Reference electrode is calomel electrode. So there is one bulb and one electrode coming. This glass electrode, glass electrode, straight.
What was the class?
See, one in coming from the glass, glass electrode, and the tip of the glass electrode contain what? Silver chloride. And another is calomel electrode. Tip of the calomel electrode contain mercurous chloride paste. So the solution in glass electrode is KCl. 7 pH KCl. So this is the electrode bulb. So for your exam purpose, this will be your spotting, in your spotting. So what is the instrument, what is the principle, what is the function, and if the examiner asks like what is the procedure, you can describe the procedure, you can tell the procedure. Okay.